Cellular Respiration for Dummies. Step 1. Glycolysis. Glycolysis literally means the breaking or splitting of sugar. It occurs in the cytoplasm where glucose is primed. In other words, 2 ATP are needed or invested in order to produce 4 ATP. The ATP are made through substrate-level phosphorylation, during which a substrate with 1 phosphate and an ADP with 2 phosphates are connected to an enzyme. The substrate hands over its phosphate to make a ATP. Also, 2 NAD plus each gain a hydrogen during this process, thus making 2 NADH. So what pretty much happened is a 6-carbon sugar was split into 2 3-carbon pyruvates, producing a net of 2 ATP along the way.
Awesome.
Now on to step 2. Oxidation of the pyruvate. First, each of the pyruvates enter the mitochondria, and there it loses an electron. Here, the pyruvates each release a CO2 and transform into acetyl groups. The acetyl groups are attracted to the coenzyme A. They get together to form acetyl coenzyme A. What's the big deal, you ask? Well, not only are 2 CO2 produced, but 2 NADH are also made, and each acetyl-CoA now has 2 carbons. Let's go over what's happened so far. 6-carbon glucose was split into 2 pyruvates, which then became 2 acetyl groups as they each released a CO2. A CoA then hopped onto each to create 2 acetyl CoAs. Time for step 3. The citric acid cycle, also known as the Krebs cycle, occurs in the inner matrix of the mitochondria.
Our drawing is obviously not as good as Miss Calder's.
During this cycle, 1 ATP, 3 NADHs, 2 FADH2s, and 2 CO2s are produced.
Well, each of the 2 acetyl groups goes through this cycle.
It all occurs twice. Thus, the end total of Krebs is 2 ATP, 6 NADHs, 2 FADH2, and 4 CO2s. Alright, here's a quick review. We originally started with a glucose molecule that had 6 carbons. In the oxidation of pyruvate, 2 CO2s were made. And in the Krebs cycle, 4 CO2s were made. So there's our 6.
Now what happened to our original 12 hydrogens? Well, glycolysis made 2 NADH, oxidation of pyruvate made another 2 NADH, and the Krebs cycle made 6 NADH and 2 FADH. So there's our original 12 hydrogens. Note that only 4 ATP have been produced so far. I know, don't freak out though. It's finally time for step 4. also known as the electron transport chain. This process occurs in a chain of proteins along the cristae of the mitochondria, which is also known as the inner matrix membrane. There, the proteins pass along electrons, causing the NADHs and FADH2s to alternate between oxidation and reduction. The passing of electrons continues until oxygen, the final acceptor, is reached. All the while, hydrogen ions are being pumped into the intermembrane. However, they need to get into the matrix, so ATP synthase gives them a tunnel to pass through. The hydrogens and ATP synthase together create ATP through a process called chemiosmosis. What basically occurs is each recycled NADH yields 3 ATP, and each recycled FADH2 yields 2 ATP. At the same time, 6 water molecules are produced. Now let's pull the whole thing together.
First, glycolysis produced 2 ATP and 2 NADHs, which electron transport chain used to make 4 ATP.
Then, oxidation of pyruvate produced 2 NADH, which the electron transport chain used to make 6 ATP and 2 carbon dioxides.
Lastly, the Krebs cycle produced 2 ATPs, 4 CO2s, 6 NADHs, which the electron transport chain used to make 18 ATPs and 2 FADH2s, which the electron transport chain used to make 4 ATPs.
So, our grand total is 32 ATP from the electron transport chain,
plus 4 ATPs from glycolysis and Krebs,
for an end result of 36 ATP. This presentation was brought to you by Calderama Productions.